Welcome to 13 Universal. And this is A-Level CA Biology. We are continuing on cell structure topicals as we wrap up the May-June 2024 series. Okay, this is your friend. This is Shedro. Okay, your tutor. All right, let's roll it. So get your writing materials. Let's solve some questions. Okay, the first one says here. Of course, you can read with me. Says a graticle and a micrometer scale can be used to measure the size of biological structures. Okay, it is viewed with a microscope. So let's look at it. So you can see this microscope. All right. So let's see the question. Which role shows the correct locations for the placement of graticle and a micrometer scale on the microscope shown? So where do we place these two? Right, we have these two scales, graticle and micrometer. So pause the video and try it. Let me see how you're doing. Okay, so if you're talking about graticle, the graticle is a scale which is used to measure specimen on the microscope. Okay, the graticle is usually embedded in the eyepiece lens. The eyepiece lens is found here. Okay, this is the eyepiece lens. This is where the eyepiece lens is, because this is the observer's lens. This is where you look through. Okay, so graticle is embedded in eyepiece lens here. So that is one. The options that have A and B, okay, are selected because the graticle is one. So, but the stage micrometer is always on the stage, so it's 3. So 1 and 3, that's the answer. Okay, B is the answer. But don't forget, this is stage. These are the objective lenses, all right? This is the coarse adjustment knob. This is the light source and all of that. So B, all right, is the answer. Let's go to the next question. 6 organelles found in eukaryotic cells are shown. Where are they? Look at this. Organelles, cute organelles. See, so they say which organelles are involved in the synthesis and secretion of glycoprotein. Okay, so these are the options. So what is the answer? Pause the video and try it. Okay, so but let me just walk you through. Don't forget that this is nucleus, right? How do I know it's nucleus? It has double membrane, this and this. Okay, this is the nuclear pores for the exits of messenger RNA made in the nucleus, exit through the pores, and then ATP or nucleotides can enter. All right, so this is nucleolus where ribosome is synthesized. This is chromosomes, right? Chromatin material is calling up, and that's nucleoplasm. So this is nucleus, guys. Number 2 is mitochondrion. Can you see? It has double membrane. Right, the outer membrane is outer membrane. The inner membrane is folded. Right, that folding is called crystal. That increases surface area for enzyme action. So the central core is matrix. This is matrix. Okay, the folded is crystal. So this is for aerobic respiration, ATP synthesis. Nucleus is for it contains genetic materials. You know that. Okay. Of course, we have this. This is centriole. Centriole is membrane, non-membrane bound cylindrical organelles. You can see cylindrical. Okay, it makes what is it called? Microtubules, which helps, all right, to move vesicles carrying molecules or move chromosomes, crystal chromatids apart during mitosis, okay, and meiosis. So you can see that is rough endoplasmic reticulum. Okay, you can see that it is interconnected membranous sacs, okay, flattened membranous sacs that on the outer membrane have ribosomes at this dark spot. So the enclosed inner cavity is called cisterna, and of course this is chloroplast. The chloroplast has internal membrane system. It has double membrane. Okay, this is a stack of membrane called thylakoid where light dependent stage occurs. That's stroma. No, so yeah, stroma is where light independent occurs. All right. So this, of course, don't forget that the function of rough endoplasmic reticulum is that it is a site for synthesis, right, transport and modification of protein. This is the site for food synthesis. All right, this is Golgi. You can see membranous sacs stacked to form a layer, so they always have, bulging ends and vesicles. So Golgi is a site for also modification, packaging and transport. So what they say, which organelles are involved in the synthesis and secretion? Synthesis first, where is it made? Okay, what is it? Where is it? What is this? How is it secreted? Which organelles are involved? Of glycoprotein. So glyco, that's glucose or chain of carbohydrate, or glucose, that's carbohydrate, or glucose, protein. So let's look at where is protein made. Okay, don't forget that protein. All right, so we are just doing revision now. So remember that protein synthesis. Remember protein synthesis. Let me use P, right, for protein synthesis is equal to translation, right? Protein synthesis P is equal to translation, TR, no, transcription first, yeah, transcription, all right, transcription plus translation. So translation occurs first, all right, sorry, transcription occurs first. Transcription is the first stage, all right, of protein synthesis. So transcription occurs in the nucleus, right? So for us to make this protein, transcription also occur here. So number one is involved. Then transcription gives rise to messenger RNA. Messenger RNA now leaves the nucleus through the spores, okay, and comes to ribosome on rough endoplasmic reticulum. And the messenger RNA is used as a template during translation. So translation occurs here, transcription occurs here, translation occurs on ribosome, okay? And then what happened here is going to be used to make polypeptide chain. Then once polypeptide is made, the polypeptide modification starts here, but modification is completed in Golgi. So Golgi is involved. So vesicles are packaged, which travels from RER to Golgi. So Golgi completes the modification process and packages in vesicles. So in vesicles, it can now fuse to cell membrane, okay, and then is released outside, okay. So but you see, so this is involved, that is involved, this is also involved. Number two is also involved because it provides energy for all of these processes. Okay, so 1, 2, 4, 6. 1, 2, 4, 6, you know it, that's B. All right, let's go to the next question. Which cell structure can have mRNA inside them? So I wanted to solve this question. Which of them have mRNA inside them? Is it chloroplast, mitochondrial nucleus, rough endoplasmic reticulum? Which one, guys? All right, any one of them, pause the video and solve it. Anyone that has mRNA, right, must have DNA. Okay, so mRNA is made using DNA. Don't forget that DNA, all right, a section of DNA which is called gene is usually during transcription. So DNA, section of DNA is copied into messenger RNA. So if we're going to be dealing with mRNA, that means that there must be presence of DNA. So the question is, the $1 million question is, which of them has DNA? So which of these have DNA? The ones that have DNA will have mRNA. Okay, chloroplast has circular DNA. Yes, this is correct. Okay, chloroplast has circular, all right, DNA. Yes. Mitochondria also has circular DNA. Okay. Nucleus has linear DNA, but it's just DNA. Okay. Rough endoplasmic reticulum doesn't have DNA. Okay. So it is 1, 2, and 3. Okay. B. Let's go on to the next question. Good. Now, I hope you are solving this question with me. A scientist carried out an experiment to separate the organelles in animal cell. Okay, that's one of them. By, all right, animal cell, and then the scientist mixed the cells with a buffer solution which had the same water potential as, right, the cells. The cells were broken open with a blender to release all the organelles. The extracted mixture was filtered and then spun in a centrifuge at a high speed to separate heaviest type of organelle. These alright, was sunk to the bottom, forming solid pellet 2. Pellet 1, sorry, that's pellet 1. So this is a liquid after centrifugation. The liquid above pellet. This is called supernatant. This is the pellet. Now this, okay, is the heaviest organelle. So after, that's the heaviest. So I have pellet 1. The liquid above pellet 1 was poured into, was drained into, alright, a centrifuge tube and spun in the centrifuge at high speed to separate the next heaviest type of organelle. This organelle sank to the bottom, forming solid pellet 2. So that's how the second one came out. The procedure was repeated twice more, okay, to obtain pellet 3 and pellet 4, each containing a single type of organelle. What is the main function of the organelle in pellet 2? Alright, so pause the video and choose the option, okay? Like you to try this, guys. Don't check the math scheme, okay? Just try it first, alright? Because this is going to be D, not 4. Alright, so if we're going to do this, let's walk through it together, okay? Alright, now remember that if what we're dealing with is plant, if it's a plant cell, we have an acronym called, okay, if it's plant, if I'm dealing with plant, I'm going to teach you for plant and animal. There's an acronym that we use to resolve this question, right? We have an acronym called nucleo, C nucleomyla. Now if it is plant, you put C, nucleomyla, nucleo, that's nu is nucleus, clo is chloroplast, myla, my is mitochondria, okay, myla, my, then let's see la, L is lysosome, okay, and the E is endoplasmic, R is ribosome. So this is from the heaviest organelle, the biggest, alright, so this is the heaviest, right, the biggest, heaviest organelle, biggest, right, to the smallest, this is smaller, okay? So that means that if we are working on plant material, this cellulose will be our pellet 1, that's the heaviest, okay? Then after that, if we drain the supernatant into the next centrifuge tube and carry out same centrifugation, the second pellet will be nucleus. So this nucleus is second heaviest, right, in plant material. The third heaviest is chloroplast, so that's going to be P3, okay. The fourth heaviest is P4, okay, and then the fifth heaviest is, that is lysosome. So P4 is mitochondrial, lysosome is the fifth heaviest, okay. All right, then the sixth heaviest is endoplasmic reticulum, that's pellet 3, no, pellet 6. Then the least heaviest, which is pellet 7, is ribosome. So guys, that is it. Now, but if we're working on animal cell, okay, we're not going to use C micro, just use nucleomyla. So if we're working on an animal cell, animal tissue, right? So our P1 is nucleus, okay, and all right. So our P2 will not be chloroplast. Chloroplast is not found in animals. So our P2 will be mitochondria. That's P2, right? Yeah, lysosome becomes our P3. And you know the functions of these organelles. So P3 is for lysosome. So that is the digestion of old organelles, okay? So P4 will be endoplasmic reticulum, okay? And then P5, pellet 5 will be ribosomes. That's P5. So guys, can you see different material determines this is pellet 5, okay? So I want you to solve this question. I've given you wings to fly, okay? The question is asking you, what is the main function? So this is the question. What is the main function of the organelle in pellet 3? And guess what? We are working on animal cell. Okay, so now I'm giving you the wings to fly. Get the question sorted. So pellet 2 is mitochondrion because it's an animal. So what's the function of mitochondrion? Is it digestion of old organelles? No, that is for lysosome, which is pellet 3. Production of ATP, yes, that's the function. But if you say production of mRNA, that's for nucleus, P1. If you say synthesis of protein, that is for endoplasmic reticulum, rough, is P4. So we're dealing with P2, which is mitochondrion, and the answer is production of ATP. So the answer should be B. All right. Which structures are found in palisade mesophyll cells and photosynthetic prokaryotes? So palisade mesophyll cell is found in the leaf, that's a plant cell, plant tissue. Photosynthetic prokaryotic cell, okay, they're bacteria. So I want you to pause the video and try this out. Which ones are found in plant cell and prokaryotes, which is bacteria? All right, pause the video. Now I want you to note that all cells have three structures in common. They all have ribosomes, they all have cell surface membrane, and they all have cytoplasm. So what I have here is ribosome and cell surface membrane. But you see, because we are dealing with plant cells have cell wall, okay? But the cell wall is cellulose cell wall, okay? Prokaryotes also have cell wall. Their cell wall is not made of cellulose cell wall. Their cell wall is made up of peptidoglycan, and then they're asking us for the structures that are found in both of them. So this is where cellulose cell wall will not be among them. If they had said cell wall, we would have considered it. But cellulose cell wall means only for plant. So we have cell surface membrane and ribosome. Okay, that's 1 and 3. Okay, which is C, Charlie. Which polymers are present in all viruses and all prokaryotes? All viruses and all prokaryotes. Which ones? Okay, so help us with the answer. Remember that all viruses, pause the video and work on it. All viruses, they consist of capsid, which is the protein that surrounds the nucleic acid. So all viruses, it could be DNA or RNA. So DNA or RNA are all polynucleotides. So polynucleotides can be found, okay, in a virus, because virus is made up of DNA or RNA. So this flies. Polypeptides, yeah, viruses have capsid, okay, and prokaryotes also have they have proteins, okay. So capsid in the virus is made up of proteins, polypeptides. So this also should be in both virus and then polysaccharide. Viruses don't have polysaccharides, okay. So the answer here is 1 and 2, which is B. Okay, then the question goes toss. It says, which combinations of lenses for a light microscope will give the greatest magnification? So don't forget that the greatest magnification is the eyepiece lens times objective lens. So what's the answer? So if you multiply the eyepiece lens magnification times objective lens magnification, the one that gives the highest, so the one that gives the highest, this is 500, okay, this is 400, this is about, if you say 40 times times 15, that gives you 600. So this is giving us, okay, 40 times 15 is 600. This is 15 times 100, which is 1500. So which one gives us the highest magnification? Okay, so that is delta. All right, the diagram shows an electromicrograph of virus particles in human nucleus. So let's look at it. This is human nucleus. All right, this is viral particle destroying, attacking this nucleus. So it says, okay, this is magnification, that's virus particle. Okay, it says, what is the diameter of the labeled virus particle? So what is the diameter of the labeled, okay, the labeled, so what you do, guys, is that in real exam, of course, you're going to be dealing with physical paper. So you go to it and then use your ruler, measure from here to here, okay? All right, so when you measure from there to there, all right, that gives you the image, all right, image distance, image height. And that image, right, if you measure it in real life, when we did it, we got 3.6 millimeters. So 3.6 millimeters. But if you convert it to, because if you look at the option, we have micrometers, of course, and nanometers. So okay, first of all, let's convert it to micrometers. So how do you convert? Of course, I've taught this before. If you're moving centimeters, you can move centimeters to millimeters. Okay, and then that's millimeters. How do you move from there to there? You multiply by 10. Okay, then you can move from millimeters to micrometers. So millimeters to micrometers, you multiply by 1000. Okay, so 1000. Okay, if you're moving from micrometers to nanometers, so this should be 1000. Okay, all right. You have also this nanometers. So we are moving from micrometers to nanometers, to multiply by 1000 again. Okay. Okay. So if we're moving from centimeters to micrometers, that's 3.6 times 1000. That's going to give us 30. All right. 3600. Okay. All right. So this is it. But we can also, if you look at the options, we have micrometers and nanometers, right? So, but if you try it, let's try it, because we have actual size is equal to image over magnification. Okay. So image is this 3600. You can, micrometers is in micrometers. Okay. You can as well convert it to nanometers. Since we have nanometer there, you can convert it. So if you multiply by 1000 again, that will be 3000. Okay. That's 6 and 3 million. Okay, 600,000. Okay, so if you bring it here, 3,600,000. Okay, so that's our image size net divided by magnification. Sorry, this is magnification, not actual. Okay, so actual is image over magnification. Magnification is 24,000. Can you see it in the equation there? 24,000. Okay, which is this? That's my question. So if you say 3,600,000 divided by 24,000, that gives you 150. Don't forget that this is in nanometers. Okay, so this will give you 1 million. Okay, so 150 nanometer. All right, so now if you check the option, which one carries it? So that 150 is the same thing as 1.5 times 10 to the power. Yeah, because if you move from here, 1, 2, that's 1.5 times 10 to the power 3. No, 10 to the power 2. Yes. So the option there is this. Okay. All right. If you have any questions, let us know. So this is the answer. Okay, let's run fast. We spent a lot of time there. It says, what is correct about the synthesis and release of glycoprotein mucin in global cells? What is correct about it? Okay, number one says the protein produced on ribosome and a carbohydrate chain is added. That's what's in the Golgi body. So to form glycoprotein, you have to add glucose to protein. So number one is correct. Number two says the glycoproteins are packed into vesicles in the Golgi body, forming lysosomes. Remember that. Of course, they say that the glycoproteins are packed into vesicles in Golgi, forming lysosomes. So lysosomes are organelles that allow digestion to occur. So this is wrong. Okay. So lysosomes carry digestive enzymes. But what we're dealing with here is glycoprotein and not digestive enzymes. So number three says secretory vesicles containing glycoproteins move from Golgi body and fuse with cells of membrane. This is correct. Okay, it's correct. So the answer should be 1 and 3. But just to paint more pictures. So if you have, for example, this is Golgi apparatus. So it's a layer of flattened membrane. Okay, and then after modification, right, then produces vesicles that carry the glycoprotein towards the cell membrane. When it gets to cell membrane, it fuses with cell membrane and then empties. Okay, so you can see from what we have there, right, you see that 2 is not correct, but 1 and 3 are correct. Okay, yeah, so you see the answer is should be 1 and 3 B. 1 and 3 B. Okay, so what are the functions of microtubules? Allowing movement of cilia in the bronchus, attachment of central medullary metaphase, moving secretory vesicles and around the cell. So what's the answer? Now if you check, guys, allowing movement of cilia, yes, correct, because microtubules form the basal membrane, basal part of cilia, so you can do that. Attachment to central, yes, so you have to attach to central during metaphase. The secretory vesicles, yes. So the answer is 3 of them. All right. Which organelle contains nucleic acids? Yeah, which of them? Golgi doesn't contain nucleic acid. Mitochondria contains circular DNA, and then of course it contains ribosome. Ribosome has of course nucleic acid. Ribosome RNA. Chloroplast contains nucleic acid. So 2 of them. D is the answer. Some features of cells are listed. You can see those features. This question keeps recurring. So which features can be found in plant cells? Okay, so you can see that's A, this is B. So I want to pause the video. Which features can be found in plant cells and in prokaryotic cells? I've always mentioned, now cell wall can be found in both of them, but just in plant cell we have cellulose cell wall, in prokaryotic we have peptidoglycan. So one can be found, the cell surface membrane is found in all cells and ribosome is found in all cells. So the answer is A. The photo micrograph shows a bronchiole and alveolar. Okay. And then I said the magnification of the image is times 360. What is the maximum diameter of the bronchial lumen? So they're asking us for, all right, the maximum diameter. All right, so we're looking for the actual. We have solved this question before, but I want you to pause the video and try to solve this question. So we're looking at, of course, actual. So actual size is equal to okay, image over magnification. Okay, so first of all, to get the actual the image, you have to take ruler, right, and measure from here to here. Now, very importantly, when we tried it, we got 5.0. We got 5.0. You can see that. So I is equal to 5.0. Okay, 5.0. When you measure it in the hard copy, but if you don't have the hard copy, you may not be able to get it exactly. So 5.04. Okay, you measure it in the one that has millimeters. Okay, so that is for millimeters. No, that's for centimeter. That's centimeters. Okay, and then of course, by the time you convert it to micrometers, okay, so if you're converting, of course, if you're converting it, we're looking for the answer. You want to convert it centimeter to micrometer times by 10,000, right? So that's 5.04 times 10,000. That gives you, okay, that I is 505, right? 1,400. So that means the actual size will now be equal to what? Okay, so this is going to be 5,400, 50,400, all over 360, which is multiplication. So when you say that divided by 360, okay, divided by 360, okay, I think there's a little mistake there. So that actually is because that thing is in, if you say 5, look at it. Okay, this is about 5.0. No, if you measure it from here to here, okay, that's 5.0 centimeter. So if you convert it, if you're moving centimeter, there's a mistake with the conversion here, right? And that's what we need to pay attention very well so don't make such mistake. If you're converting 5.0 centimeter to micrometers, if you're moving from centimeters, right, to micrometers, you have to multiply by 10,000, right? Because you have to first of all convert to, right, millimeters by 10. So if you move centimeter to micrometers, multiply by 10,000. Yes. So if you say, if you see now, this is going to be by right. Okay. So what we're going to have here is going to be beyond that. It's going to be more than that. So we're going to have is 5.04. Okay. 5.04 times 10,000. Yeah. Okay. That will give us what? 5.04 times 10,000. That gives us, right, 5.04. Okay. Now, once you have it, so of course you divide it by 360. Yeah. So I think we're right from the first place. Okay. So divide by 360. So we're right. So this should give us 50,400. Is that okay? So you put it here. 50,400 divided by 360. Did you get? Yeah, that will give you 140. So that means the actual size is 140. Okay, 140 micrometers. Yeah, 140 micrometers. That's it. All right, and that is C there. Okay, let's run. A specimen is observed twice using microscope. Let's look at it. Is observed twice. A microscope firstly using green light with a wavelength of 510, and then using red light with a wavelength of 650. So what happens to the magnification and resolution when red light compared, right, to green light? Okay, so pause the video and try to solve it. Okay, so it was initially this. Pause the video and try it, and then they now moved it to this, right? So what happens? Remember that this deals with wavelength, okay? And don't forget that wavelength is indirectly proportional to resolution. So that means that the shorter the wavelength, the higher the resolution. So wavelength is indirectly proportional to resolution. That's the relationship there. So the one that has a shorter wavelength will have higher resolution. So that means that 510 nanometer will have higher resolution. Why? Because it's shorter, has shorter wavelength. 650 will have low resolution. Okay? So wavelength deals with resolution. So what happens is, can you see, if you move from 510, basically, if you move from 510 to 650, you are moved from high resolution to low resolution. So that means that resolution will decrease. So let's check the one that has resolution decreases. That's this. Okay? So this is high resolution because it's shorter wavelength. This is low resolution. So moving from 510 to 650 is moving from high resolution to low resolution. Because the shorter the wavelength, the higher resolution. Okay? The one that has shorter resolution wavelength will have higher resolution. Okay, so that's a decrease. And of course, nothing happened to magnification. Magnification remains the same. See. All right, 4 students were asked to match the function with appearance of some cell structures in an animal cell. Look at it. These are the functions listed by number. The first one produces mitotic spindles during cell division. 2, synthesis of polypeptide. 3, synthesis of lipids. Okay. So, see the appearances. So we're going to match it. Okay. So this is a simple question. This number 1 says produces mitotic spindle during cell division. Which organelle produces mitotic spindle? Central. Okay, which one describes central? V says membrane which surrounds. Now central doesn't have membrane. W says no membrane bound spherical. Yeah, this is ribosome. Okay, W describes ribosome. X is a double membrane interspersed with pores. This describes nuclear membrane. Okay, Y, V describes either rough or smooth and reticulum. Okay, Y is a non-membrane bound cylindrical structure. Yeah, this is now central, and central makes mitotic spindle. So 1 is Y. So let's come here. Which one is Y? Okay, C and D has 1 as Y. So eliminate A and B. Okay. Now synthesis of polypeptide synthesized in the ribosome. Which one is ribosome here? W. Okay. So you come to C and D. Okay. 2 is W, because synthesis of polypeptide is done by ribosome. Ribosome is non-membrane bound spherical. So you see, both ribosome and central, they are both non-membrane bound. But it's just that ribosome has spherical structure, while it has cylindrical. All right. So of course, the last one says synthesis of lipids, which is smooth and reticulum. V is the answer. All right. So if you check the option, you see that the option answer is there looking at us. Okay. So which students correctly match the numbered functions with appearance of the cell structure? Okay. See, student C did the job. All right. What is it? Okay. Found in chloroplast and mitochondria. So what is found in them? Pause the video and try it. Okay, so both of them have 70S ribosome and circular DNA. That's B. Which feature is correct for all known viruses? You see, they keep repeating this question. All known viruses, right? They say capsid made of lipid and protein. Capsid is made up of lipid only. Yeah, all known viruses are made up of course, they have capsid. But I want you to pause the video and try by yourself, okay? Now they're all made up of capsid. I hope you know what is called capsid. I'm taking time. Okay, basically, viruses, usually have. You see, this is capsid. Okay, beads or protein, right? Joined together. Each bead is a capsomere. So you can see this is capsid. Now this capsid is called protein coat. So now inside this is a nucleic acid. So they're made up of capsid, but that capsid is not made up of lipids. It's protein. So that's what is called phase A. Okay. B says DNA core. Not all viruses have DNA. Some have RNA, nucleic acid. Okay, as genetic material. So that also eliminates B. RNA, okay, or DNA. That's the genetic material of virus, RNA or DNA. So outer envelope of phospholipid, not all of them have outer envelope of phospholipid, but all of them are non-cellular. They have non-cellular structure. Okay. So non-cellular structure means they do not have organelles. All viruses don't have. They lack organelles. They lack of course cellular structure. So that's why the answer is D. Okay, diagram shows the state of micrometer with division 0.1 millimeters apart. Viewed. Okay, let's look at it. Okay. Yeah, apart, viewed through an eyepiece containing graticle. So you can see that. Okay. Alright, they say the area of the field of view can be calculated using this formula. So this is how to. So that's a student calculated the area of field of view of the microscope using the information provided. This is that. Which answer has been rounded correctly to an appropriate number or single figure? Okay, so we need to get the answer first. So guys, pause this video and try the questions. Alright, so but the first thing I want to say is that look, this is a state micrometer. We use state micrometer, these thick ones, to calibrate eyepiece. This is the eyepiece graticle. The eyepiece graticle is used for measurement, while state is used for calibration. Okay, they already told us here the diagram shows a state micrometer with division 0.1 millimeters apart. So from here to here is 0.1, from here to here is 0.1 millimeters. But for us to calibrate, we have to find point of overlap. Point where the eyepiece coincides with state micrometer. Now this is a point of overlap here. This is a point of overlap. This is another point. We're going to choose two. So if you choose this here and here, you can see that from here to here is one state micrometer. But from here to here is how many eyepiece graticle? Okay, lowest is 10, highest is 50. Okay, so you say 50 minus 10 will give you 40. So from here to here, because from here to here is 10, 20, 30, 40. So 40 eyepiece graticle gives us 1 SM. Okay, don't forget that 1 SM is 0.1 millimeters apart. So let's get into the game now, guys. Come swim with me. All right. So don't forget that this is 0.1 millimeters. You can convert it to micrometers because the answer is in micrometer, okay, and millimeter too. But we want to convert it. If you convert it from millimeter to micrometer, you multiply by 1,000. Okay, so times 1,000. All right, that's 10 to the power 3, and that gives you 100 micrometers. So we have 100 micrometers representing, all right, 1 eyepiece graticle. Okay, so don't forget that we said that 1 SM, okay, 1 state micrometer, all right, gives us 40 eyepiece graticle. Okay, all right, yeah, so we have established it. And that 1 SM is the same thing as 100 micrometers. Is that okay? Because 1 SM is 0.1 millimeters. They said that with divisions 0.1 millimeter apart. So converting it to get 100. So I can say that in another way, that 40 eyepiece graticle represents my 1 SM. Okay? My 1 SM is equivalent to 100 micrometers. Okay. All right, 100 micrometers. Yeah. So we're not much interested in calibration here. Is that okay? Not much interested in calibration, right? They said we should calculate area. So what we're looking at is area. But you see, to calculate area, you need to find radius. Okay? Alright. So the radius of this graph is from the circumference to the center. That's the radius. This is our radius. So radius represents how many eyepiece graticle? 50. Right? From, you can see it from here to here is 10, 20, 30, 40, 50. That's radius. Alright? So 440 eyepiece gives us 100 micrometers. So 50 will now give us what? That will give us our radius, okay? Yeah, 50 will give us what measurement? So it's going to be 50 cross and multiply 50 times 100 divided by 40. So you say 50 times 100. All right, 50 times 100 divided by 40. All right, what will it give you? So get your machine and just say 50 times 100, okay, divided by 40. That's 125. So that means that our radius there, okay, is 125 micrometers. That's what represents 50. 125 micrometers. All right. Okay. So our area now, remember this is micrometers. This is micrometers. Sorry for. Right. So our area is pi. If you check this out, area is pi, which is 3.142. Okay. Times radius. Radius is radius square, 125 square. All right. So get your calculator and punch it. So our area will now be equal to what? Area will be equal to what? Okay. So let me punch it. So 125 squared. 125 squared is what? Okay. 125. That's let me do it again. 125 times 125, that's 125 squared. 125 squared is 125 times 125. Okay, that gives you 15,725. But you see, times 3.142, which is pi. And that gives us, yes, so this gives us, of course, 49093, okay, 0.75. Okay, so we're looking at, all right, significant figure. So if you turn it to standard form, from here, this is 1, from that point, 2, 3, 4, 4. All right, so this is 4.9. But to make it stand significant figure, all right, the 0 is not significant figure, so we'll cut 91 and put it there. 4.91 times 10 raised to the power 4. Okay, and I don't have to go for that. You know the answer. You can see the answer there. Can you see the answer there? The answer is delta. Is that okay? That's delta. All right, D. Okay, you can watch the video again and follow and ask questions if you have any question. So let's look at the next question. You say, where are cisterna found in a cell? Pause the video and try it. Where are they found? Where can I find cisterna? Now cisterna is enclosed, okay, fluid, a closed cavity, and it's always found in endoplasmic reticulum and Golgi, 2 of them. It's not found in mitochondria. So 1 and 2 is A. Which row matches the functions of the cell structures? Okay. Which row? So vacuole stores water, sugar, that's correct. Nucleolus manufactures messenger RNA. No, it doesn't manufacture messenger RNA, manufactures ribosome. So A is eliminated. B, Vacuole export protein, does not export protein. C. Stores water, sugar, and pigment. Yes, that's correct. Manufactures ribosomal RNA. Yes, that's correct. Ribosome protein synthesis. Yes. Lysosome contains enzymes. Yes, of course. The answer is C. If you check D now, vacuole doesn't export proteins, and of course, ribosomes does not synthesize lipids. All right. So the answer is C. All right. So which structures are found in prokaryotes and eukaryotes? You know that just structures that are found in all cells, whether it is right prokaryotes or eukaryotes, ribosomes, cell membrane, and of course cytoplasm. No, nucleus is not found in all cells. I think it's B. Okay. Yeah. All right, guys. Thank you for hanging on. We're going to be doing another topic on biological molecules. So watch the next video on biological molecules. See you. This is Shedro. Right. Bye.